Hello everyone, my name is Ahmed. Thank you very much, Louis, for the introduction. It's a real pleasure and an honor to be here to speak to you today. So we're going to be talking about causal modeling agents, which is a framework that we are creating at the UCL Center for Artificial Intelligence. So very briefly, my name is Ahmed. I'm a clinical doctor by background. I did 2 years of practice after medical school and then became interested in AI. So I did a master's in machine learning at Imperial, and now I'm a Microsoft PhD scholar at UCL. And I'll let Deji introduce himself quickly.
Hello everyone, as Ahmed said, it's very much a pleasure to be talking here today. So I'm Ayodeji, also known as Deji Ijishakin. I'm also a PhD student at UCL up the road, focusing on generative modeling and applying that to diseases like Alzheimer's disease and ALS disease.
Okay, so let's start with a general problem statement, and it's relatively straightforward. So it's given a data set which is potentially multimodal, so maybe you have some unstructured text data, maybe you have an Excel file with lots of columns of numerical data and some images. Can you work out a set of causal relationships between those variables? So the basic idea is I throw some data in, maybe some images and a CSV file, and I get out a graph which describes the relationship between the variables. So this causes that, which has some downstream effect on this other thing, and so on and so forth. In this talk, we're going to present a system which does this. So this task is called causal discovery. So it's a system that performs causal discovery by synthesizing large language model capabilities with causal modeling. Now, to help ground intuition for the talk, we're going to have like a running example application, and the one we've chosen is Alzheimer's disease. So obviously the UK has an aging population. Alzheimer's is already a bit of an issue. Global prevalence is projected to be about 100 million by 2050 and 1.6 million in 2040 in the UK alone. So it's an increasingly important thing to be thinking about. And as a complex neurodegenerative disorder, its etiology, that is to say, the causal explanation of the mechanism of the disease, is deeply contentious and broadly unknown. So it's a perfect candidate to kind of challenge ourselves to try and work out what's going on. Broadly speaking, we're going to be using the framework of causal machine learning, which is a big umbrella term that subsumes lots of many different things. In our case, it's the use of machine learning to try and tease apart structure from its empirical implications. Broadly, though, we're going to use causal machine learning for three big things. The first is causal discovery, as we mentioned. So from unstructured data, how can we derive causal structure? Once you do have a model, though, and you've trained it on a data set, you can perform interventions. So you can say, what's going to happen to me in the future? So this can be used for prognostication or disease progression modeling, for example. And the third type of thing we're interested in at the bottom there is treatment effect estimation, which is utilizing something called counterfactual inference. So these are sort of retrospective hypothetical experiments that you can't actually do scientifically. So a good example in the sense of Alzheimer's disease could be, if I was born biologically female, what would my disease process have looked like? Obviously, that is currently outside of the scope of science, but can be done from a computational point of view under certain assumptions. So what we're going to do is introduce you to the CMA. So the CMA does three things broadly. So it designs a hypothesis, and its hypothesis is embedded into a modeling space. It then fits this model to some data, and then it can iterate its hypotheses. It can self-improve its own ideas, depending on some sort of model fit. Now, we're going to leverage large language models to help us design hypotheses, and a model class called a Deep Structural Causal Model, which I'll kind of decompose and talk about momentarily. The Deep Structural Causal Models, or DSCMs, as I'll refer to them from now on, are the data engines of our agent. They can fit models to data that you have, so that you can run experiments afterwards. And the LLMs, we view them as metadata-based reasoning engines. Now, when I say reasoning, that can trigger all sorts of interesting philosophical debates about whether LLMs can generalize or not, and do they actually reason or not. Although interesting philosophically, we're slightly less interested about that in this particular talk. So when we say metadata-based reasoning, all we're saying is that we can use relevant context to have useful outputs that is not based on numerical data in any way. So to understand deep structural causal models, I'm going to mention some causal graphs, normalizing flows, and VAEs, and then pass on to Deji. So I guess let's get started with that. The first thing is the concept of a causal graph, which is relatively straightforward. Supposing you're trying to model 5 simple variables. So here we've got age, sex, ventricular volume, which is a fluid-filled sac in the middle of your brain volume itself, and X at the bottom there, just imagine that that stands for MRI brain image. Now you can see, for example, in the top left that age influences B for brain volume and V for ventricular volume, and that brain volume and ventricular volume then influence the phenomenology of your brain MRI scan. Now, this is a relatively straightforward structure, and I want to show you something that looks a tiny bit more complicated, but it's not more complicated, it's just the same thing, and I'll explain it slowly. So this graph on the right looks a little bit scarier, but actually it encodes the exact same causal relationships as the graph on the left. But the only thing that this graph allows us to do, the only reason I put it here, is that we can explain some assumptions slightly more clearly about how we model these types of graphs. So the first thing is that the graph has to be acyclic, and acyclic simply means that we cannot ever return to a node. So if I start at node A here and I go to B, there's no way to sort of, no matter where I go, I can't get back to A. This acyclicity assumption is fair enough because many processes in nature are temporal evolutions of a natural experiment. So you don't want to go against time anyway, though it's not perfect, and we'll mention that briefly towards the end. The other thing to note is that there are all these kind of weird new nodes called epsilon, like epsilon B for brain volume. And if I just focus on that momentarily. So Judea Pearl, a very famous computer scientist who sort of really formalized counterfactual inference, came up with this idea of sort of seeing these nodes in gray as endogenous variables. So these are things that you really see in the world. So if I measured your brain volume, this is this node B. Now, of course, there are so many unmeasured confounders. The world is stochastic and random, and we don't actually capture everything in science. So he just sort of represents that with this epsilon term, beta. And this bidirectional arrow here, this white and black-headed arrow, is meant to represent an invertible function. That is, some way to be able to go from our actual observations to the sort of exogenous noise, to the state of the world that is compatible with it. And if we have an estimation of the state of the world, which is epsilon b, we should be able to use that exact same function to get back to the endogenous state, so to predict what the brain volume should actually be. Now, okay, we have a challenge now, which is we create a graph. We want to model these relationships, and when I say model these relationships, I mean each arrow in the graph I showed you earlier, that needs to be a deep neural network. We want to model this flexibly, but ideally it needs to be invertible. For low-dimensional things, we're going to use these things called normalizing flows, which some of you may have heard of, and if not, don't worry, I'll touch on it very briefly in a minute. And for complex, high-dimensional objects, we're going to use a type of generative model called a variational autoencoder, a VAE. Now, let's start with the normalizing flows. So let's just take the example of ventricular volume V here. This epsilon term, I'm going to assume is just Gaussian noise. You might be wondering why Gaussian, why a bell-shaped curve? It's just a design choice, and it's just very common in the machine learning literature. And we want to learn some complex function that can predict from a bell curve the actual observed ventricular volume of some patients with Alzheimer's disease. So here's how we'll do it with a normalizing flow. If I can ask you to look at the left of the slide, you'll see that we've got this red node Z0 here, and you've got this bell-shaped curve. And what normalizing flows are, they're a type of deep neural network that composes lots of small functions together. You have function 1, then 2, then 3, then so on, all the way up to function fk. And what happens is that each one of these functions will mold the Gaussian curve, the bell-shaped curve here, into something more complex. And when I say something more complex, what I really mean is it molds to your actual data distribution in the real world, which can be arbitrarily complicated. One important thing to note is that you can reverse these functions. Once you learn them, you can go from the noise, the state of the world, to your data. And if you've got some data, you can go back to the state of the world that is compatible with it. So that's in a nutshell the normalizing flow. We're on the home straight. We have all the pieces we need to define a CMA momentarily, but just one other thing is the variational autoencoder. And again, many of you will be familiar with this, and if not, here's a very quick sort of exposition of it. So the idea is we take brain MRI images, we put them into a neural network called an encoder, which produces a low-dimensional latent, a low-dimensional summary of the MRI image. But what's actually happening here is that we are predicting the parameters of a multi-dimensional bell-shaped curve Gaussian distribution. And what we do is we take a random, so here, just for interpretability, I've plotted a two-dimensional Gaussian. What happens is we take a random sample from this Gaussian, so this yellow dot. This is our data point. We take this data point, we put it back into the decoder, which is a different neural network, and it's tasked with reconstructing the image. We do this so that in this sort of middle blue zone, we learn a latent hidden representation of brain MRI images. For technical reasons that are outside of the scope of this talk, you need to sort of wrap this entire process into another invertible function so that you can, go to the state of the world, back to your endogenous noise. For anyone that's interested, you can come and chat to us afterwards. But the main point to understand, though, is that we can make this computationally tractable computation using a VAE. So now I'll pass on to Deji to just explain the method of the causal modeling agent with all of these parts.
Thanks a lot, Ahmed. So Ahmed touched on how we're able to model the different dependencies within a graph. But I guess the next question is, how do we incorporate LLMs into the process so we can discover novel relationships, including variables within the graph and those outside of it? So the causal modeling agent is decomposed into 4 parts, as you can see. Each part is taken up by a panel. And the first part is the global hypothesis amendment phase. And, a bit tricky. Okay, so the first part is the global hypothesis amendment phase, which is the first row. And it starts off with an input graph. Then there's a memory, which I'll elaborate on when we get to the final panel. And then you ask an LLM to output a graph based off of that. So because it's an iterative process, it's going to continuously happen. We're going to go in the loop, looping all 4 parts until we meet a convergence criterion. So you can imagine in the very first step, all we have is a set of variables. And we ask an LLM, how do you think these variables should be arranged? Once we get an output from the global hypothesis amendment phase, we then encode that DAG as a JSON representation. And then we move on to the local hypothesis amendment phase, which is the second row. At this point, we begin to consider all the individual variables within the DAG and all the different relationships, and consider whether we want to amend them, i.e. remove the relationship, introduce an associative as opposed to a causal relationship, or make some other form of amendment. And at this point, we have the option to use RAG, which Louis was touching on before. So you can imagine for a given relationship, there's going to be some disease context which is related to it, and therefore can allow the LLM to make a better judgment. So an example would be if you're looking at the relationship between a given gene and someone's age, a gene like the other way around, someone's age and a gene like AV45 in the context of Alzheimer's disease is going to have a lot of associated literature that is useful for the LLM when evaluating this relationship. And out of the local hypothesis amendment phase, we then get another DAG, which again we represent through a JSON representation. Once we finally have this JSON representation, we can build a computational graph using this JSON, and then we can fit our deep structural causal model that Ahmed was describing earlier to this graph. And this will output a final graph and a likelihood. The likelihood is telling you how well does this graph describe my data? What is the fit of my model to my data? And once you have this, we go to the final stage, which is the post-processing stage. We give the LLM the graph and the likelihood and any changes which had happened a step before. And then we get the LLM to give us a synopsis of what's going on. So you can imagine if I make a particular change, let's say I flip an arrow between a gene and some other brain biomarker, then the likelihood could go up or the likelihood could go down. And what we want to do is get the LLM, or we can say the same, to be fair. But what we want to do is get the LLM to describe, okay, why do you think this has happened? Put it in the context of the process that I'm evaluating. And to take a step back, what we've ended up with is one stage where we've asked the LLM to produce a hypothesis, then we fit a model to that hypothesis, then we get some feedback, and we just iteratively go through this process. And that's the basic idea behind the CMA. So let's look at some results. So although our main use case or main application would be Alzheimer's disease, we also benchmarked our approach against various different traditional causal discovery algorithms on 3 different data sets. So if you look at the first column, well, there's various different algorithms, and these are numerical approaches to discover the true DAG given a set of variables. And the idea is they use asymmetries within the variables you have in order to uncover how the different dependencies exist within the data. And if we go across the next few columns, we have 3 different data sets. So the first column is the Arctic sea ice data set, which is a DAG which describes the relationships between Arctic sea ice variables and the North Pole. The second one is an Alzheimer's disease DAG, which we produced in collaboration with neurologists at UCL to describe the etiology of Alzheimer's disease. And the final data set was the Sangiovese data set, which describes the grape purity of different grapes in Tuscany. So there's different ways that you can measure how good a DAG is. One way is the normalized Hamming distance. You can think of the distance of your DAG from the true DAG you're trying to uncover. Another one is the baseline Hamming distance, which you can think of the distance of your DAG from the worst case scenario. But if you take the ratio of it, then it's a less biased measure because the normalized Hamming distance is contingent on the number of edges you have. And for the ratio, the lower it is, the better the DAG you have. And if you look at our results, you can see that we outperform the traditional numerical methods when it comes to fitting DAGs to these various different data sets. And we also outperform just GPT-3 and GPT-4 by themselves when trying to do causal discovery on these DAGs. And what that's kind of a implicit ablation study in that it shows that the iterative process that we employ with the CMA is actually giving a performance gain. So let's move on to some more results relating to Alzheimer's disease in particular. So what we have here are image counterfactuals. And as Ahmed had described previously, a counterfactual is when you intervene on a variable within a DAG and then see how the downstream effects of that intervention produce different results. So it's essentially asking the question, how would the world have been if something were different? And I'd like to direct your attention to the final row. So what we're doing in the final row, what we're doing in the final row is intervening particularly on ventricular volume. So the ventricles are a fluid-filled sac in the middle of your brain. And if you look at the original brain image here, this person has a 23.87 ml ventricular volume. And immediately to the right, we're intervening on that image and increasing their ventricular volume to 150 ml as opposed to 23.87. To make that very clear, you can look at a difference map. Here, red means lower pixel intensity values. So it suggests that within that fluid filled sac, the intraventricular volume, there is more black space, i.e. a higher ventricular volume. And you can see if you do the reverse at the very end of that panel, if we try and make someone's ventricular volume smaller, you can see the inverse change, i.e. there's a contraction of the ventricles. But something that's very important to note is the digital identity of this person is the same. If you look at the other components of the image, i.e. the sulci and the gyri or the cortical thickness, these things remain the same, whereas only the variable that we're caring about changing actually changes. And you can imagine the impact of this for the clinical research within Alzheimer's disease. So those were some results on what you're able to do with the model once you have it. But what we really care about is discovering the etiology of Alzheimer's disease. How does this process work in the end? So this is an example DAG which comes out after the process. And it looks very complicated. There's all sorts of relationships which may not be necessarily very interpretable if you're not a neurologist. So the question is, okay, how good is this DAG? How do I know it's any good? So I'm going to pass back to Ahmed so he can explain how we're evaluating the quality of our DAGs.
Yep, thanks, Deji. So, as Deji said, so this is a DAG that comes out from our trained model. And the way that we benchmarked this was we spoke with 5 experts from the Institute of Neurology. Now, 3 of these experts are professors of clinical neurology and 2 of them are professors of clinical neuroradiology. All of them have a subspecialist interest in neurodegenerative disease or Alzheimer's. And it's tough to see here, but you could see it quantitatively in our ratios that we quoted earlier. Our model basically does very well. So every edge that it predicts is kind of causally sensible and kind of biologically plausible. And I mean, don't worry too much about the names, but I'll just give you an example of like, so EDU is like educational attainment across however many years you've been educated. MOCA is the Montreal Cognitive Assessment Score, which is kind of an IQ test, but specifically in the context of trying to measure degenerative changes in the brain. You have some genes like AB45 and ApoE4 and so on. One important thing to note, though, is that our CMA actually managed to propose things that none of the experts proposed. It's hard to sort of see, but this top left node here is biological sex, and this node here is something called phosphorylated tau, which is a protein that causes damage to the human brain. And our model predicts this red edge here, which says that biological sex has an effect on phosphorylated tau. This was the optimal model that came out of our CMA, which was surprising because we'd never kind of thought about this before. You just have to take my word for it. Neither did the neurologists. So we were like, well, okay, you know, none of these experts have kind of said that this was a sensible hypothesis. That might be because there are super linearly many relationships to consider, and it's difficult for a human, even when you have like 12 variables, to consider every possible DAG. There's more than 10 billion DAGs, in fact, to consider. But it could be that they did think about it, and they didn't think it was biologically plausible. So we kind of dug into this, and because the DSCM that I mentioned earlier trains on data, we can run experiments. So we can say, okay, we're going to take, on the left, all the men and counterfactually set their status to being women, which is, as I was mentioning earlier, a biologically kind of impossible experiment to do, but you can do it in data. And on the right, we took all the women and counterfactually set their status to be men. And the most important thing to note is kind of, I don't know, is my mouse visible? Yes, it is. So is kind of these top distributions. So in red are men and their level of phosphorylated tau, this dangerous protein. And in dashed blue are women, which you can sort of see in both instances, there's a sort of transposition to the right. We tested this. This was very, very strongly statistically significant. There's many thousands of patients here, so even though the difference doesn't look huge, it is quite big. And what our model seems to have said is that women have a higher level of phosphorylated tau. Now, dig as we might in the literature, it's not completely well delimited, this relationship, but there is emerging evidence of it. So there was a paper published quite recently in Cell in October 2022, which found this gene called USP11, ubiquitin-specific peptidase 11. And it's on the X chromosome, and women have 2 of them. And it escapes something called X inactivation, which means women have a higher expression of this particular gene, and it basically seems to increase the level of this degenerative protein when they have Alzheimer's. So we were really kind of excited by this, and we dug a little bit deeper into what other sorts of things the CMA could do. And because we use LLMs to design hypotheses, there's no reason to stop at the bounds of your current data. So we'll give one example here. When it was looking at a possible relationship between variables we do have, so this ApoE4 is a gene that increases your risk of Alzheimer's, and this is phosphorylated tau that we've been discussing. So the LLM puts out this like sentence, and basically the long and short of it, it's like, you know, other genetic factors such as TRM2 could potentially confound this relationship. And again, this is a really nice way of modeling data because we hadn't heard of TRM2, so we were like, well, what is that? So we actually went and collected TRM2 data, and we added it to the graph, and then we ran additional experiments, and we found that if you increase the soluble version of TRM2, it's called sTRM2, you have higher levels of this degenerative protein. And this was doubly interesting because we really, truly, to the best of our knowledge, cannot find the mechanistic pathway for this. So we know that TRM2 is an immune receptor. We know it's something to do with the central nervous system. It's broken down to its soluble form, sTRM2, by this protein called Adam metalloproteinase. Fine. That's kind of all we know. So our work proposes a potential relationship between the soluble form and tau deposition. But as of April 2022, which is the most recent meta-review on this topic, it's just unclear what that relationship looks like. We don't know if it's the soluble form has a biological impact or somehow breaking it down matters. But again, this is very interesting because what we're trying to show you here is that our framework can propose insightful relationships, both in the variables that you do have access to, so where the experts missed between sex and tau, and variables you might not have access to as interesting and important confounders that you should go and collect, which is a powerful paradigm when you consider just modeling numerical data by itself. So for final conclusions, I'll give it back to you, I think. Yep. Yeah.
Thanks, Ahmed. So to conclude, we presented the CMA, which is a modular framework for causal discovery using LLMs and DSCMs. And the real advantage that we would like to postulate about this combination is that when using LLMs, we're trying to mimic how human beings, such as expert neurologists, would reason over these variables. And that has many advantages because it allows you to prune away like unsensible relationships, but also allows you to use your expert knowledge to introduce novel relationships. And DSCMs obviously have their modeling advantages. And by bringing both of them together, you kind of have something which tries to mimic an automatic scientist up until the point of actually carrying out those scientific investigations. So as a result, the CMA was able to propose several insightful relationships, which were both internal and external to the data engine. And this is something that Ahmed had just briefly touched on. The example of an internal novel relationship was the relationship between sex and p-tau, which was taken out or unteased from the data by our modeling engine in combination with the LLM. And something which was external was the T-REM2 relationship. And we were able to demonstrate that these relationships may even be beyond the current expert consensus. The idea would be a model like this should be like a handmate to a scientist trying to come up with new hypotheses if they have a set of data. Of course, there are some important limitations that we have to address. One of them is the Markovian assumption. So the Markovian assumption is required in the current form of modeling causal relationships because we use DAGs and the certain conditional independent statements that DAGs have. And what this means is they can't be cyclic relationships. However, in nature, there are cyclic relationships. So for example, the way that the stock market is affects the news about the stock market, and the news about the stock market affects the way that people think about the stock market, but the way that people think about the stock market affects the stock market. So there's a cyclic relationship between those variables. With a model like this, you wouldn't be able to model that. However, this is something which is still an open question about how we can incorporate this into causal modeling in the first place. So although we're not addressing it, it's definitely something which should be considered in future work. The final thing is to address the Markov equivalence class limitation of our approach. So you can think of a Markov equivalence class as a set of graphs which make the same mathematical statements, the same conditional independent statements about the probability distribution that they're modeling. And you can also, a very intuitive way of thinking about it is a set of graphs which have the same general structure if you look at them from a high level view. And our model, as well as numerical models, can only bring you up to a Markov equivalence class, i.e. if the true graph exists within this group, then our approach can get you up to the group but not past it. However, there has been recent work by Long and colleagues which has demonstrated that if you start off with the Markov equivalence class, then you can use large language models to get to the true DAG. And the intuition here speaks again to the point that we were making about trying to mimic the way that human beings model these types of relationships. Because if I have a set of graphs with the same mathematical statements, then some of them may just be implausible. A good example would be if with one of those graphs said that how old you are is affected by your gender, for instance, I immediately would remove that as a human being, but a numerical method wouldn't have any sensitivity to that. So I think, again, that's something that we're going to be considering in future work about addressing these limitations. But thank you very much. Thank you so much for listening. Hope you've enjoyed it very much. And yeah, open for some questions.